Hello, my name is Monika Laux. Welcome to my presentation about nickel phytomining efficiency of different hyperaccumulator species in Austria. I did my master thesis as part of this project, and my supervisors were Markus Puschenreiter and Christina Hipfinger.
Let's start with some explanations. Hyperaccumulators are plants that are able to take up huge amounts of trace elements, especially metals, from the soil and accumulate them in their above-ground biomass. They have to meet these criteria. The shoot concentration has to exceed a certain threshold. For nickel, the threshold is 1000 micrograms per gram. The concentrations have to be higher in the plants than in the soil. That means the bioaccumulation factor exceeds 1. Furthermore, the metal is translocated from the root to the shoot, and the shoot concentration is higher than the root concentration. Thus, the translocation factor also exceeds 1. The hyperaccumulator plants don't show any toxicity symptoms, as normal plants would certainly do with these properties. Therefore, they can grow on metal-enriched soils, like serpentine soils. On the pictures, you can see some example for nickel hyperaccumulator plants. The first plant is Berkea codii, a plant from South Africa. It was one of the first plants known for nickel hyperaccumulation. Odontaria chalcidica is a plant from Southeast Europe. It has been investigated in many studies before. Phyllanthus bolgioi is a tree that grows in Malaysia. As you can tell by the green color, its phloem sap has a very high nickel concentration. It's about 15%. But there are also local nickel accumulator plants. Nocea goesingensis is one example. Phytomining is the technology to recover the metal from the hyperaccumulators. After growing and harvesting the metal crops, they are burned to recover the energy and to receive the ash. The ash is then a bio-ore, and it has a higher nickel content than mined ores. The ash can then be washed and the nickel can be leached with acids. After further purification, there is produced a nickel salt. A second possibility is to use the bio-ore as a starting product for a smelter process and to obtain the nickel metal. Agromine is a term that was introduced in 2015. It covers the whole process chain from plant production to metal recovery. LIFE-Agromine is a project that is funded by the EU and intends to demonstrate the phytomining efficiency of 4 different locations of Central and Southern Europe. The study sites are located in Albania, Austria, Greece, and Spain. In France and Belgium, there are other parts of the study conducted. For example, a full life cycle assessment of cropping hyperaccumulators for nickel recovery. And there also investigated the associated ecosystem services like soil improvement. The project duration was from 2016 to 2020, so we are in the last year, the year of evaluation. My master thesis is embedded in the Austrian part of the Agromine project. My research questions are the evaluation of the phytomining efficiency of 4 different hyperaccumulator species, which are Odontaria chalcidica, we have heard about it before, Bonmüllera tymphe and Leptoplax imaginata, which are 2 Brassicaceae from the southeastern Europe also, and Berkea codii, which is the plant from South Africa. As we are in the 3rd year of the project, I tried to evaluate the long-term efficiency of phytomining with Odontaria chalcidica. And I tried to investigate the effects of phytomining on the soil characteristics. Following the results of my predecessors, The biomass is the decisive factor for increasing the nickel yield. According to literature, von Müllerer, Leptoplax and Berkea, the new species of this year, might have higher nickel concentrations and therefore exceed the nickel yield of Odontaria chalcidica. So we will apply the agronomic techniques that have been optimized in the last 2 years.
Here you can see an aerial view of the study site. It's called Redelschlag, near Bernstein, and it is located in the serpentine region of Burgenland in the very east of Austria. Here you can see the plot for the comparison of the species, which was the main part of my master thesis. Here on top you can see the old plots, which were planted in former years. 4 of them were replanted in 2019 to evaluate the long-term efficiency of phytomining on this site. Here you can see an upscaling plot, which was evaluated by Christina Hipfinger.
Here you can see the experimental setup of the comparison of species experiment. There were 4 plots for each of the mentioned species. In addition, there were 4 plots that were kept unplanted as a control for the effects on the soil quality. And there were 4 more plots that were planted with Odontaria chalcidica but were not harvested in September but in April, to see if the yield could be even increased if letting the plants grow over the winter. But these were not part of my master thesis. Each plot has 10 square meters, was fertilized with pig manure, and in each plot was planted with 4 plants, so we have a plant density of 4 plants per square meter. To evaluate the long-term efficiency of phytomining on this site, 4 of the old plots were replanted with Odontaria chalcidica. Just as the other plots, there were 10 square meters and there were 40 plants per each plot. They were not fertilized this year.
In 2019, the plants were harvested at the beginning of October, over 3 consecutive days. Here you can see another scheme of the study site and another aerial view. The old plots and the new plots for the comparison of the species, which were part of my master thesis, and the upscaling plot. We produced 1 mixed soil sample for each plot, with 12 punctures on each plot. We took the soil samples down to the depth of 12 centimeters. From each plot, we took 4 representative plants as samples. The plants were taken from specific positions on each plot.
Here you can see some more impressions of the harvesting days. This is myself with one of the biggest plants that we harvested. Here you can see the plots with the different species. Leptoplax, von Müllerer, Berkea and Odontaria. We had help from some very nice students and we could pack a lot of vans full of plants. Back in the lab in Tulln, we could dry all our samples. To dry the plant samples took quite some time as we had harvested so many plants and the capacity of the greenhouse was really too small for all these plants. Well, after that we could start the analysis. The chemical soil parameters we determined were the calcium exchange capacity in the barium chloride extract, the available phosphorus with the Olson method, the pH, the CN ratio, the easy available nickel in the strontium nitrate extract, and the potential long-term plant available nickel pool in the DTPA extracts. The biomass of the plants was evaluated as an average for each plot. The CN ratio was measured. The plants were digested in nitric acid and hydrogen peroxide, and the digests were analyzed with ICP-MS and ICP-OES. Here you can see the results for the above ground shoot biomass. This year Odontoria chalcidica was growing really well with a biomass of nearly 6 tons per hectare. This is more than in the years before. Bornmüllera and Leptoplax didn't even show half as much biomass, and Berkea was even significantly lower.
The result of the plant nickel concentrations. You can see that Bornmüllera and Leptoplax tend to show a higher concentration than Odontoria chalcidica, just as expected. But the difference is not significant. Only Berkea codii shows a significantly lower nickel concentration in the shoot biomass.
If we compare the nickel concentrations to the other plant nutrient concentrations, we see that the green bars, which are the nickel concentrations, are comparable to the calcium concentrations and are even higher than other macronutrients like phosphorus or magnesium.
Finally, when we combine the nickel shoot concentration and the shoot biomass, we receive the nickel yield. As you can see on the left bar, Odontorea chalcidica has the highest nickel yield, about 94 kg per hectare. Bornmüllera and Leptoplax have significantly lower nickel yields due to the lower biomasses. Berkea codii combines the lowest nickel concentration and the lowest biomass, thus the lowest nickel yield.
Finally, we take a look at the long-term efficiency of nickel phytomining on this site. We see that in the first 2 years, the nickel shoot concentration do not differ significantly. Just in the third year, 2019, we see a significant increase in the shoot concentrations, which means that the nickel phytomining efficiency does not decrease over 3 years.
But what about the effects of phytomining on the nickel concentrations in soils? The label nickel fraction decreased over the growing period in the plots of Odontorea and Bornmüllera, and there was no measurable effect on the control plot. And what about the long run? Well, we saw the tendency that the label nickel pools get replenished after the growing period over the winter. That is an indication that the phytomining efficiency will not decrease for quite a few years. Conclusions that we can draw from our results. The nickel concentrations in the plant shoots were the highest in Leptoplax and Bornmüllera, just as expected. But Leptoplax and Bornmüllera did not even produce half the amount of biomass per hectare compared to Odontorea. Thus, the highest nickel yield was achieved with Odontorea chalcidica, reaching 94 kilograms per hectare, which is not as much as in the domestic areas of Odontorea chalcidica, where you can achieve up to 200 kilograms per hectare. In contrast, Berkea codii showed the lowest nickel concentrations and the lowest dry weights of shoots, thus it did not meet our expectations. Our results therefore show that Odontorea chalcidica is the best performing nickel phytomining plant species for the Austrian site in Bernstein. And for this species, we also found that the nickel shoot concentrations did not decrease within 3 years. But the nickel soil concentrations tend to decrease from plantation to harvest, but they replenish afterwards until the start of the next growing period. This is the list of my references, which are really just the classics of phytomining. So, thank you for listening. Hope to see you next time face to face. Goodbye.